Biological discovery is a process of building on earlier achievements. Several important steps led to the discovery that DNA holds the hereditary information. Let's look at how biologists first determined that chromosomes are the mediators of inherited traits. In the late 1800s, Gregor Mendel, an Austrian monk doing experiments with pea plants, discovered that specific traits are determined by inherited factors, later called genes. Mendel's work went largely unnoticed until the early 1900s, when American biologist Walter Sutton and German embryologist Theodore Boveri independently noticed that the behavior of chromosomes in egg and sperm development exactly matched Mendel's theory of inherited genes. This led them to believe that Mendel's genes are located on chromosomes. They proposed the Chromosome Theory of inheritance, which stated that chromosomes carry genes, the hereditary information. The chromosomal theory of inheritance was further supported when Thomas Morgan was able to figure out that the gene that gave fruit flies red eyes was the next groundbreaking experiment in the discovery of DNA as the genetic material was the first bacterial transformation, performed by British medical officer Frederick Griffith in 1928. Bacterial transformation is the process by which bacteria are changed by the uptake of foreign DNA. But at the time of Griffith's experiment, nothing was known about bacterial transformation. Griffith was studying the bacterium Streptococcus pneumoniae, which Griffith found that the smooth cells were able to kill injected mice, while the rough cells could not. When he used heat to kill the smooth cells, they were no longer able to kill the mice. However, when heat-killed smooth cells were mixed with the non-lethal live rough cells, the injected mice died. And live smooth bacteria could be recovered from the dead mice. Somehow, the rough cells had acquired the ability to make the polysaccharide coat and were now able to kill the mice. At the time, Griffith didn't know what made the rough cells become smooth cells. However, his experiment gave other scientists a starting point to look for the nature of the genetic material responsible for the transformation. In 1944, American bacteriologists Oswald Avery and Colin McLoud used the data from Griffith's experiments to determine what the transforming agent was. They purified various substances from bacterial cells and tried to transform rough bacterial cells with each of the substances individually. Avery and McLoud found that only DNA was able to transform the bacteria and make the bacteria deadly to the injected mice. The rough cells picked up the smooth cell DNA